cambias al inglés.
Cambias de idioma.
Entonces hago la presentación en inglés, pero si ustedes tienen preguntas, me pueden hacer en español, ¿ok?
Ok.
Okay. Bien, voy a colocar mi presentación para ustedes verán. Un minuto.
Estas cosas.
Estoy a cargar mi presentación. ¿Ustedes ven mi presentación ahora? ¿No?
Se vio un momento. ¿Ahora?
Ahora sí.
¿Ahora sí?
Sí.
No.
¿La ven?
Sí.
Ok, perfecto. Voy a colocar aquí. Bueno, entonces voy a hablar un poco de la validación y la aceptación de los métodos alternativos. I don't know if Pilar already presented me. I have had many years of experience in the validation of alternative methods and in the regulatory implementation of the alternative methods here in Europe. It's more than 20 years, and before this I did a little bit of development of alternative methods. Now I work in a 3R center that was created in Switzerland to promote the 3R principles. But I will be talking to you mainly about the validation of alternative methods and their acceptance. Okay? So just a little bit of background, maybe you already heard, but in the 80s, there was a lot of ethical concern about the use of animals for experimentation, and there was a lot of movement from the animal welfare groups. This is an example of a homepage of the New York Times that was financed by this man, Henry Spieren, about the use of rabbits for the cosmetics testing. And this triggered a lot of things in Europe. One of the things was the directive on the protection of animals. Oh, sorry, I made a mistake. No, sorry. I tried to reduce this. So the use of the directive for the protection of animals used for scientific purposes that I guess you already heard about. And in particular, it says that if the member states shall ensure that if a scientifically satisfactory method or testing strategy that does not use a live animal should be used instead of the procedure. The procedure meaning the animal experimentation. So there was a real push towards the validation. This is checking the scientific validity of the method. This was implemented in '86 and updated in 2010, but this article remained the same. What was added in the update was that it's not only the method, but also a testing strategy. So there was already the notion that you may need more than one method to achieve the replacement of the animal testing. And at the same time, in the 90s, there was a lot of studies that were undertaking to try to replace animal testing. This was one of the first studies that took place, and it was for eye irritation and serious damage. There was one study from the Commission, from the COLIPA, which is the Cosmetics Association, from Germany, from the USA, 2 different studies, from Japan as well. Altogether, more than 30 in vitro methods were tested, more than hundreds of materials were tested, and there was a huge number of laboratories. And these studies, they thought that they could replace the whole animal testing with one method, and they found that this was not so easy. And unfortunately, the studies were not successful, but there was a number of lessons learned that developed the main concepts of what we know today as validation study. So Michael Bowles, for example, was involved in these studies, and what he did is he profited from everything that didn't work and tried to make what do we need to do that this validation can work. And this gave rise to this workshop report in '95 about practical aspects of the validation toxicity test procedure, as well as this task force report on pre-validation, so what you need to do before you start the validation study, that was written by Roger Curran, also in '95. And these principles give the basis of what we know today as the principles of validation study that were integrated in 2005 and OECD guidance document 34. And this is basically the principles that today are still used to validate alternative methods. And this has been accepted by the 36 member countries of the OECD. Okay. So what is the validation process about? So the validation is to establish the reliability, so that means the reproducibility and the relevance of a method for a particular purpose. It's important to keep in mind the purpose because it may have different validity for a different purpose. So if you have here our alternative test method and here the reference test that can be human, that can be animal, then what we will check is the reproducibility of this alternative method as well as the relevance. And the relevance is, in one hand, how well it predicts the reference test, and on the other point of view, the scientific basis of your method. And this relevance is what distinguishes the validation of these alternative methods from the validation of analytical method. So what is an alternative replacement test? It can be regarded as a combination of the test system and a prediction model, which is an explicit decision-making rule for converting the results of one or more alternative tests into a prediction of an in vivo pharmacotoxicological endpoint. And this is a little bit the validation process. So we go from research and development to the pre-validation, the validation, the peer review, and the regulatory acceptance. And it requires this collaboration and communication between all the players that are involved, the academia, industry, the validation bodies, and the regulators. To enter the validation process, there is a set of minimum criteria that is usually required. So, for example, the definition of the purpose of the method, as I mentioned before, is very important. The mechanistic basis of the method, why it is a relevant method, and then the availability of an optimized protocol, which can include standard operating procedures, specifications of the endpoints and endpoint measurements, how a preliminary prediction model. So a prediction model can be like, for example, very simple. If a toxicity is higher than 50%, then it means that it will induce skin irritation, for example. It's a very important step in defining the protocol. And nowadays we see more and more complex prediction models, but that are very important to predict the reference test. And then an indication, if possible, of the preliminary applicability of the test method. So perhaps this is a method that was developed for a specific class of chemicals or for a specific mechanistic prediction. And this is what we define as the applicability domain. There can be different types of validation study. You may have a prospective validation study, which represents an inter-laboratory study, but usually you have at least 3 laboratories that take part. And these laboratories will test a number of substances, some together, some may be not the same, but you have to have a statistically justified number of chemicals to allow to assess both reproducibility and the predictive capacity of your method. Then you can have a retrospective validation study based on the weight of evidence. So that takes into account the existing data that are already available of your method, and this is combined together to see how reliable it is. And then you may have a catch-up validation study, which is in the case that a method has been already validated, and there is a new method that is developed that is very similar to the validated method. So we call it a me-too assay. And then it doesn't need to go to the whole process of validation, but one can make a a much smaller validation study, usually based on what we call the performance standard. So we define the performance, the minimum performance that this similar test need to fulfill for them to be considered valid. And these performance standards are usually published at the OECD level, for example. This is a little bit the structure of how a validation study looks like. So we have the sponsors who pay for the study, a management team who will define the goals of the study and the design of the study, and will be responsible for the coordination and the approval of the protocols and the evaluation. And then we have the laboratories. So usually we have a lead laboratory, which is the laboratory that developed the assay, and two other laboratories participate, or in this case, for example, you have a lead laboratory for one test, and then two other participating laboratories. And in case you have more than one test method being evaluated, then you will have a lead laboratory for the second test and the lead laboratory for the third test and the participating laboratory. Very important is to have an independent group that will do the selection of chemicals that do not comprise any representative of the laboratories that are testing. And then also very important is to have an independent biostatistician that will help in defining what is the minimum number of chemicals that is needed to have a statistically justified sample of chemicals tested. In 2004, Thomas Hartung defined what we call the modular approach to validation, which define exactly what are the information that you need to obtain. So we have the test definition, then the within laboratory reproducibility of a method, the transferability in a next laboratory, and the between laboratory reproducibility. So this is the reliability part of the validation. Then we have the predictive capacity. So how much of the positives your method can correctly predict, or how much of your negatives your method can correctly predict, and the applicability domain, and together they will define the relevance together with the scientific basis of your method, and the performance standards that are defined for these Me-Too assays that I mentioned, so similar assays. And when all of this information is provided, the modules are completed. This is then reviewed by the validation management group, and following then an independent peer review. The general principles of the validation are that for a method to be judged valid, it has to be reliable, which means reproducible, and relevant, so predicting the right thing, because it doesn't have to have a very reproducible test but does not predict what we want to see. It's important that the prediction model is defined in advance by the test developer, so before the validation study takes place. And the performance criteria should also be set in advance by the management team. The validation is conducted with coded chemicals. And as I mentioned before, there should be independence in the management of the study, in the selection, coding, and the distribution of the chemicals, and on the data collection and statistical analysis. Ideally, all the laboratories that take part of the validation study should comply with the GLP or GCCP principles. When the validation study is finished and the results are considered satisfactory by the validation management group, then this is peer reviewed. In Europe, it is the ESAC who peer review the validation studies. We are having, for example, a meeting next week with ESAC, the new ESAC that was formed. These represent the new ESAC were nominated last year. I am part of it. There are 9 core members and additional ad hoc external scientists. And we are coming from different backgrounds: academia, industry organizations, public institutions. And I guess the independent consultant was me, but I became now a public institution. This will perform then independent reviews, and it will publish ESAC opinions. In the past, this used to be the ESAC statement. These ESAC opinions are then used by ECVAM, the European Center for the Validation of Alternative Methods, to make test method recommendations. And this is a little bit a summary of how it looks like. So if there is here the validation study, it undergoes peer review by ESAC, and then ECVAM makes the recommendation. And when ECVAM makes the recommendation, it consults with the stakeholders. It can be the stakeholders from the regulatory agencies, which is called parere, or it can be stakeholders that have an interest on the validation of alternative methods, and this is called the ESTAF. But it also consults with ICATAM. ICATAM is the International Cooperation on Alternative Methods, which is comprised of validation bodies from the United States, from Canada, from Japan, from Korea, and from Brazil. The idea is to align the principles of validation between the different parts of the globe. Then ECVAM publishes the recommendation for public commenting. It takes into account the public input and publishes the final recommendation. And this was the process that was established by Joachim Kreser, who was also director of ECVAM. And what I mentioned to you up to now was mainly developed for chemicals, but if you check in the area of pharmaceuticals, recently, well, already a few years ago, the EMA published a guideline on regulatory acceptance of 3R testing approaches, which is applicable to human and veterinary medicinal products. And we can see that the criteria that they established for regulatory acceptance is same as what I have mentioned to you. So the OECD guidance document 34, which is also the principles applied by ECVAM, the European Center of Validation of Alternative Methods, and by ICVAM, which is the American equivalent of ECVAM. But in the pharmaceutical area, it also proposes two other options. One is in case that there is a recognized gap, that it is possible to have a case-by-case evaluation by national control authorities. So there is a discussion between the data submitters and the authorities that if they can demonstrate that the alternative data is as useful as, if not better, than those using the existing methods. And another possibility that is given is what they call the safe harbor process, which is a process that conducts in parallel the testing with the new method and the testing with the previous method. And the data is used by the authorities only to compare how well one predicts the other for some time before the new method is applied. So in the pharmaceutical area, there is this principle that I mentioned to you, plus these two other cases. And there is also a guidance for the transfer of methods between laboratories in the case of the 3Rs. So today, maybe you already heard before, but so I will pass very fast over it. But the requirement for alternative methods are multi-sectorial, and that is why it's gaining more and more importance. Well, I think you have heard, Pilar must have told you about the cosmetics directive, where this is no longer possible to test ingredients using animals. But also for chemicals, we have some areas where only in vitro testing should be conducted, for example, skin and eye irritation and skin sensitization, unless the chemical is outside of the applicability domain of the in vitro methods. And in particular in skin sensitization, Europe has implemented the mechanistic approach, which I will describe more in details. But what is also very interesting is that in this REACH regulation for chemicals, there are different levels of acceptance depending on the validation status of your method. And I think this is very important. So if you have a method that is validated, the regulators will accept it for identifying hazardous effects, but also non-hazardous effects. Whereas If your method is suitable means that it fulfills the minimum criteria to enter the validation study, as I mentioned earlier. In that case, the method can be used only for identifying hazard effects. But in any other assay can also be used for gaining mechanistic insights. This is a little bit how the recommendations are. For example, for testing skin and eye irritation is first to take into account all the existing data, physical, chemical, human data, existing animal data, QSAR, existing in vitro data, and do what we call a weight of evidence analysis. If this is sufficient to classify, we classify. If not, we go for the in vitro method. And if only when the in vitro studies are not applicable because the substance is outside of the applicability domain of this method, then we would go for an in vivo testing. So it's really, the in vivo is using really as a last resort. And as I mentioned, for skin sensitization, it is very nice because we have in the legislation now implemented really a series of in vitro and in chemical methods that tackle different key events of skin sensitization, the molecular interaction with skin proteins, the inflammatory responses in keratinocytes, and the activation of dendrocytes. Again, the in vivo study should be conducted only if the in vitro methods are not applicable because they fall outside of the applicability domain of the method. Then we have the legislation for mixtures, where also here there is a distinction between validated and non-validated method, and where the tiered weight of evidence strategy, as I mentioned before, is encouraged, as well as biases. And finally, the same with pharmaceuticals. In 2017, EMA has published a number of opportunities for limiting animal testing that can be already implemented. So as you can see, it's not only in one sector that these alternative methods are needed, but really overall in different regulatory scenarios and in different sectors. We do have some alternative methods that are already available for regulatory purposes. This is just an overview of the different methods. And by the way, I can give you the PDF presentation of a copy of my presentation afterwards. And the same for the pharmaceuticals. There is a list of regulatory guidelines that can be amended or implemented with PCR methods. This includes, for example, biologicals, biosimilars, safety guidelines, immunologicals, vaccines, guidelines, and efficacy of veterinary product. And here I will give just some examples for you, but if you want to have further information, I would suggest to take a look in the previous slides that I gave in the specific websites. So for replacement in Europe today, one example is the skin irritation and corrosion. In the past, we had one in vivo method that was the OECD test guideline 404, and this has been replaced by 4 in vitro test guidelines, 1 for irritation and 3 for corrosion. That represents 10 different test methods that you can see here. And there are additional ones that will be included here, additional 2, and additional 1 implemented here. So we will have a total of 13 test methods. And then the regulators were a bit puzzled, but how can we combine these tests? How do they work together? So in 2009, the OECD started a project to see how do we need to combine these different guidelines. And there was a guidance document published on the integrated approach to testing and assessment for this purpose. It tackled different modules, as I mentioned before, the human data, the animal data, the in vitro data, non-testing data like physical chemical properties and QSARs, as well as a guidance on the weight of evidence analysis. And it also described the applicability, the benefits and limitations of the existing test guidelines and modules. For the first time at the OECD level, in this guidance document, it was possible to identify non-hazard effects if conducted with a validated alternative method. So it was no longer only first corrosion and then irritation, but also the possibility to start with first irritation. And if it's negative, consider as negative and then go for corrosion. And there was also a discussion about the usefulness of the test guidelines for mixtures and preparation. The guidance document was adopted in 2014, and all the test guidelines were adopted accordingly. And this is how it looks like. So you have three main parts of this guidance document. Part one is everything that relates to non-testing methods. So it's the existing information, human, in vivo, in vitro. The physical-chemical properties, for example, pH, can already give indication on the corrosive effects. And the non-testing methods like read-across, grouping, QSARs, or in the case of mixtures, the bridging principles and theory of additivity. And then all of this information is combined together in a weight of evidence approach. If this is sufficient, we classify and label, and if not, we go either to what we call a top-down approach in case there is more likelihood that this is an irritant or corrosive, starting with a method for corrosion and then a method for irritation, or a bottom-up approach in case there is not a high likelihood that this is irritant. Then we start with a method for skin irritation, and then we do the skin corrosion, and we classify according to the outcome. It's important to say that this replacement is only valid in Europe, because in Europe we only have one category for irritation, which is the category 2, and then the category for corrosive and non-classified. Some countries may implement a category which is called mild irritation, and in that case we will need certainly additional methods to do. But what is very nice, and I think maybe this may be adopted even next month, at the United Nations level, they are now changing the definitions of these classification categories, what we call the globally harmonized system for classification, to include already the classification based on in vitro methods, because so far this was only based on the in vivo observation. So that is really a change that is occurring very broad. Then another example that I wanted to give is a partial replacement method, and this is exactly about the first slide that I showed about the eye irritation and corrosion studies. Today, the in vivo test, which is the Dre's rabbit eye test, we have a number of methods that were validated to identify no need for classification, and a number of methods that were validated to identify serious eye damage, so corrosion. But we don't have anything to identify the mid category, which is the irritation. And why is that? Because for eye irritation, there is a lot of concern regarding the safety of consumers. When the regulators wanted to have methods to identify serious eye damage, they wanted to be sure about the results, so have very few false positives. But when you decrease the number of false positives, what happens is that you increase the number of false negatives. So we do have a number of false negatives in some of the assays. And the other way around, for the identification of the chemicals that do not require classification, for the safety of consumers, this was very low, 0%, 1%, 2%, the maximum 4% of false negatives, to be really sure that the chemical that is not identified here does not cause any effects on the eyes. But then in counterpart, you have a big number of false positives. So in the end, in the mid category, we will have a high number of false negatives category 1 and a high number of false positives no category, and we need to identify this. And in particular here on this end of the scale for serious eye damage, why do we have this big number of false negatives? We found out that a high percentage of the chemicals that we have evaluated, so this is number of databases that are available on chemicals, at least 56% were classified as causing severe eye irritation, not because of acute severity of effects, but because in the animal test, in the rabbit, these were effects that lasted for 21 days. The problem is that most of the methods that were adopted so far for this endpoint do not evaluate the reversibility of effects, but more acute effects. So that's probably one of the reasons why we have this high number of false negatives. However, in some areas, it is now possible to find methods that may predict the reversibility. This is one example. It's histopathology of the isolated chicken eye. So in the isolated chicken eye, you take away the eye of the chickens entirely, and you do the test. And in addition, you can preserve the eyes and do histopathology. So this is, for example, the histopathology of a control cornea. And this is what the effects that we found that correlate well with persistent effects. One is the erosion of cornea that is very moderate. So at least half of the cornea, the part of the cornea is gone. Or another effect that we found that seems really to be correlated with inducing serious eye damage is these small vacuoles that are formed, but only when they are formed in the lower and mid layers, suggesting that then this is not reversing anymore. And we found that when we use the histopathology, we can predict much more of these persistent chemicals than if you only use the acute test. So this has been now included this year, actually in July, if I remember well, in the OECD test guideline, and the guidance document explains how to do this. Also here, there is a need for an integrated approach for testing and assessment, because here again, we need to combine the different assays, and it's very similar to the skin one with 3 parts: the existing data, non-testing data, the weight of evidence, and then the in vitro testing data. Sorry. And what is very interesting in this IATA document is that for the first time, we have considerations on the intrinsic characteristics of the animal test, and also talking about non-OECD adopted method. The IATA was adopted in 2017, and I wanted to give you a little bit of insights on the animal test, because often we think that the animal test is a good test and is the perfect test, especially regulators have a tendency to think so. But in the case of the Drey's eye test, so the substance is applied in the cul-de-sac here of the eye of the animal, and the eyes are closed for 1 minute. Then the eyes are, if this is a liquid substance, not washed for 24 hours. And depending on the type of substance you have, for example, if this is a solid material, it may cause abrasion to the eyes. The solids are washed after 1 hour, but still it may cause abrasion of the eyes. And if this is a hydrophilic material, it will leave faster the eye. And if this is not hydrophilic, then it may remain longer time with the eyes. If you have a glue, for example, it will be there for a long time. So actually, we don't know the real duration of exposure, because if this is washed easy with tears, then it's fast. But if it not, it stays there in the eye quite some time. There might be variation on the animal responses because of the blinking and lacrimation. And in the case of solid material, as I mentioned, because of the mechanical abrasion, that could lead to a secondary inflammation that is not caused by the chemical itself, but just because of this mechanical damage. And finally, there is subjectivity in the allocation of the respective scores. And the variation that can occur with the animal test has been quantified. And we see that the concordance of classification between experiments, so we are talking is within laboratory variation, is in the order of 60%, which is huge. And we also saw that from within experiment, there is 11% of chances that a serious eye damage material is under predicted as causing irritation, and 12% that an eye irritant is under predicted as not needing classification. So this is only within test. And finally, it was found that there is 10% chances that a non-classification is given before a serious eye irritant, so corrosion. So 10% chances of mixing no classification with corrosion. So it's a very variable assay, and we very rarely put in the question the animal assay, and the animal assay also has its drawbacks, and it's important to bear this in mind. This is a little bit the list of the non-OECD methods that are included in this IATA guidance document, and many of them are now under discussion at the OECD level, but very interesting is to see that there are also new methods that are being developed to try to detect this persistency of or irreversibility of effects in the eye. Porcora, for example, is an assay that uses porcine cornea and that are cultivated for 21 days, just like the rabbit eye test, and allows to see if there is a kind of a reversibility or non-reversibility of effects. And then the third and the last example I would like to give is about adverse outcome pathways and the use of defined approach to testing and assessment. And this is the case of skin sensitization. In this case, there is a very clear defined adverse outcome pathway where after skin penetration, the protein is binded and it induces keratinocyte responses. So this would be the protein binding, the key initiating event. Then a key event 2, which is the keratinocyte responses, and a dendritic cell activation as a key event 3. And this then leads to dendritic cell migration, dendritic cell and T cell interaction, T cell proliferation, the creation of memory cells that will then result in the adverse effects. In the past, we had mainly refinement assays for this assay, which were the reduced LLNA and different variations of the local lymph node assay, which is the LLNA. But more recently, we have validated test methods that cover these 3 different events. So one is the direct petite reactivity assay. So this, if you remember, is the test that I mentioned that in Europe is now based on the mechanistic approach. And this is the OECD test guideline 442C. Then we have for the key event 2, the keratinocyte activation, which are 2 methods, keratinosense and lusense, which is called the key event test guideline 442D. And then we have a key event test guideline for the key event 3, which is about the dendritic cell activation. And here there are 3 methods: the HCLAD method, the USense method from France, and the IL-8-LUC assay from Japan. So as you can see, in this area of skin sensitization, also here, you will need to use methods in combination to predict whether there is an adverse outcome or not. But it's not like in the case of eye and skin irritation that you will use them in a tiered approach one after the other. Here, you also need to use them in combination. And that gave rise to the definition of what we call a defined approach to testing. So the defined approaches are an integration of readouts of a suite of in silico, in chemical, and in vitro methods. And in contrast to the IATA that I mentioned to you before, the defined approaches are based on a fixed set of information sources, and it has data interpretation procedure. For example, there is one defined approach that says that if 2 out of these 3 tests are positive, then you know that is positive. If 2 out of 3 are negative, then you predict this is negative. This is just one example. But in 2016, there was a guidance document published on the definition of the defined approach, where there were 12 case examples, case studies of possible combinations of the assays, and that allow to predict not only the hazard for skin sensitization, but perhaps also the potency. And at the moment, there are 3 of them that are under discussion to include in a test guideline on defined approaches. And we have a meeting also next week at the OECD level to see how this will proceed. But it's a very, very interesting work that is being conducted. So, well, I gave you the 3 examples, and now, before I finish my presentation, I would like to share with you what I believe are the future challenges and difficulties that we still have ahead of us, and some possible prospects. One of the things that I think is very important is, like in the case of the skin sensitization, the adverse outcome pathway. So this is the sequential chain of causal linked events that lead from macromolecular interactions, cellular responses, organ responses, until you have the full organism response. Nowadays, it's possible to propose an AOP to the OECD, and this is the link where you can find this information. And the OECD has developed what we call the AOP knowledge base, an interactive web platform on AOPs. And there you have AOPs that are available for commenting. There are some AOPs that have been already peer-reviewed, some that are under review, and some that are under development. But I think this is a very important source to go more and more towards mechanistic predictions. In 2016, the EPAA, the European Platform on Alternative Methods, which combined the European Commission and a number of industry sectors, made this publication, a workshop that resulted in this publication that was published recently, where it identified the challenges for further implementation of alternative methods. And in particular, one of the main challenges found was the further characterization of the pathways of toxicity, but also how they interact. And when we have characterized this very well, is to develop the assays that cover the scientific gaps that currently exist. It is also important to better characterize the link between the in vitro readouts and the final outcome on the target species. So what will be the consequences of these in vitro readouts, and a better definition of the alternative methods applicability domain. But altogether, the main constraint that this workshop identified is really that there are still some existing gaps in the scientific knowledge and in technical limitations that do need to be overcome. The regulators also hold a workshop in 2017. So there was a workshop organized by BFR from Germany and RIVM from the Netherlands, including regulators, the OECD, government, industry, and academia. And also here they say that the way forward would be to step away from the animal study as the gold standard. I mentioned to you that there are some drawbacks, and also there is interspecies variation, and define what is the human biologically based regulatory requirements for human hazard and risk assessment. So here again, going to much more mechanistic pathways. And in 2006, I published together with Maurice Whelan, who is director of ECOM, this book about the validation of alternative methods, where the main challenges identified are summarized here. And in this pathway-based approach, what we see as a major hurdle is to moving from an information that is qualitative about the mechanism to quantified, and that there is a need to be flexible to validate based on these modes of action, and that you don't need only cellular assays or the mechanisms associated to it, but you also need extrapolation models extrapolate from what you observe in vitro to whether this is something that has a real consequence in vivo or not. And for example, in the case of transcriptomics, it's important to see what is the threshold that shows that really there is no more return, a threshold of adversity. In the case of stem cells, it's important to better standardize these models, and there is a number of elements that need to be very well characterized, like identity, sources of variability, the quality, the characterization of the pluripotency, the characterization of the differentiation, the functional potential, the stability and window of use. Whereas for bioreactors, he suggested that the validation is based on process characterization, so like quality by design, rather than doing a validation at the end of the process. And finally, regarding human ownership and microphysiological system, here we talk more about qualification of the system, evaluating the reproducibility, the cellular system, how to combine different growing cells, the stability in time, the organ functionality, and the standardization of testing protocol. Recently, just this year, I think last month or 2 months ago, the OECD published a new guidance document, which is called the Good In Vitro Method Practices. This is a guidance document that was developed by ECVAM and the OECD together, a process in consultation with a number of stakeholders, which aims to provide guidance on the development and implementation of in vitro methods for regulatory use in human safety assessment. In particular, for example, it provides these 10 chapters that describe the different steps of what is important to take into account when someone wants to develop a method that might be useful for regulatory use. So it discussed the responsibilities of the different players, as I showed in the first slide. It talks about quality considerations regarding the management, the quality control, documentation. It talks about the facilities, the apparatus, materials, and reagents. It talks about the cell test systems, how to characterize the different cells that are used, the identity, the stocking, the cryopreservation. It talks about test and reference items, including the controls that are needed to ensure that your new method provides reliable and reproducible results. It talks about the standard operating procedures, the test method performances, which is what I mentioned about the validation of these methods, the reliability, the acceptance criteria, and the demonstration of proficiency. And it talks about reporting of the results and the storage and retention of records and materials. And finally, but not least, there was a guidelines published in 2014 describing what, in case you have an in vitro method that is not adopted by the OECD, what is the information that needs to be provided on this method. This was mainly developed for novel in vitro assays like high throughput screening and complex model. And basically, these are very similar to the information that I have provided so far regarding the test method definition, the purposes, the principle, the quality and acceptance criterion, the known limitations and strengths, the prediction model again, which is very important, the performances regarding predictive capacity, reproducibility, scope and limitations, and the potential regulatory applications. Some more example, is it more used to support a read across for priority setting? Is it more useful for screening purposes, or is it proposed as a component of the IATA? So there are a number of tools that are being developed to allow to progress in this towards this more mechanistic view of the alternative methods. So in conclusion, what I can say is that the use of alternative methods is important because of ethical, regulatory, but also scientific needs. And that is multi-sectorial, is not limited to one sector, but is applied to different sectors. The validation was important to facilitate the regulatory acceptance. And today we do have some areas where we have full replacement, some areas where we have partial replacement, and some areas where we only have reduction and refinement. This is the survey from ECHA that was published last year, if I remember well, of the chemicals that are registered in the European Union. Out of 6,290 substances submitted for registration, 98% were submitted through data sharing. So this is already allowing to reduce the number of animal testing. 89% of these substances had at least one endpoint based on alternative to animal testing, and the alternative that was most frequently used was the read across, followed by weight of evidence and then QSAR. Sorry, 20% of the dossier for the lower tier endpoints contained in vitro studies, and the lower tier endpoints include skin corrosion, irritation, eye hazard, acute toxicity, genetic toxicity, short-term for skin and eye hazard, and have multiplied of 6.6 between 2011 and 2016, representing from 400 studies in 2011 to almost 3,000 studies in 2016. Interestingly to see is that more than 56% of the studies were conducted in vitro, more than 50% were based on existing in vivo data, and there was only 4% of in vivo studies. So there is really a reduction of in vivo studies. Regarding in skin sensitization, the number of in vitro and in chemical tests were multiplied by 4 between 2011 and 2017, and I'm pretty sure this number will increase in the next report considerably. What is also very interesting is that because most of the guidelines were adopted around 2015 and 18, so I'm pretty sure this will increase. Most of the applicants make use of this integrated approaches to testing and assessment, and only 11% of the requirements were covered by new animal studies. But we also have a number of challenges that still need to be of a tendency to be a little bit conservative and to consider the animal as the gold standard over human effects. And the second challenge is the length of the validation and the acceptance process. But we also have some opportunities going towards this more mechanistic approach, the use of these defined approaches and diatas, and the scientific progresses, because today we are able to revert the stem cells to make them pluripotent and transform them in any cell type we want, and that is a great resource to be able to use human-based models. And there is more and more development of 3D models and organoids and the human on a chip. So I believe that in the recent years there has been a great scientific advances in this area, and combined together with this more mechanistic approach and validating and checking the quality of these new methodologies, I really think we can do a lot of progress. So this is it. I thank you for your attention, and don't hesitate to make me questions if you have any questions.
Chantra?
My camera to see you.
Can you see me? Hi?
Can you see me or?
Yeah, a small one. You are here, very small, only. Okay. Any questions? Well, I think people is very tired now, and it was a very excellent talk, as I usually do. And thank you for participating in this course. Obrigada, Chantra.
Nada. So, if you have any questions, you can contact me. Me pueden contactar cuando quieran, que puedo ayudar con las preguntas.
Muchas gracias. Tendremos la presentación, ¿no? Porque hay mucha información.
Sí.
Okay. Thank you.
Okay.
Bye.
Buenas
Bye, bye.
noches entonces para ustedes.
Y para ti también, ¿no?
Gracias, gracias.
Chao, chao.
Chao, chao.
Bueno, pues damos por acabado el curso. Ya habéis visto que lo que habíamos estado hablando de órgano on a chip y lo que ha comentado Rosavilla de la importancia de la validación ha salido. Y varias cosas que hemos estado hablando han salido, todo lo de la validación. También ha hablado de lo que yo os había comentado esta tarde de las buenas prácticas de laboratorio en toxicología in vitro. O sea que esta ha sido como un poco el colofón de todo lo que ha sido este curso, y porque es importante, si no hay validación no sirve para nada sustituir los métodos por otros métodos. Y bueno, hay mucho trabajo por delante. Bueno, gracias por vuestra asistencia y espero que os haya gustado el curso y que haya cumplido vuestras expectativas.